So my job here is to lay the foundation for the rest of the panel. Just because not all of us are clinical researchers and some of the terms are unfamiliar. And so we're going to start with the basics. We might, there we go. What's a clinical trial anyway? Clinical trials are the final steps in new therapy development, and they're defined by involving human subjects who are treated with an intervention by a protocol to measure a health-related outcome. And as we heard yesterday, there's important regulatory oversight of that process. And this is different from other types of clinical research that happens in humans, perhaps that's observational, or registries and things like that. We learn a ton of information from that type of research, but clinical trials is really how we move forward with therapeutics. Having a little trouble with my button up here, you guys. Okay. So this diagram shows the steps in therapeutic drug development. And although it's important to know that clinical trials, although it is important to know that clinical trials can test other therapies and interventions that are not pharmaceutical related, the process, I want you to know, often takes a really long time, well over a decade from start to finish, and to be able to use those therapeutics clinically. They can cost hundreds of millions of dollars or more for just one drug to be available in for clinical use. So we sort of see the end, the tip of that iceberg, and it's on that side here, but not all of the pieces that have to go into it. And so I often get questions about things that are in the preclinical phase that somebody's read something about, super interesting, really thought-provoking, may go on to be a clinical trial, but there's a long way between that preclinical research and actually using that therapy. So it starts on the left-hand panel here with some sort of idea, moves through to a preclinical testing phase in a lab with animal models, tissues, and then if the idea gets that far and does well, then it goes into human studies. Phase 1 studies are our smallest studies. They're the shortest, and they're usually looking for safety and dosing. You've heard this earlier in the weekend. Phase 2, those studies are a little bit bigger, but not all of our phase 1s make it to phase 2 because they're unsafe or something like that. They take a little bit longer, and we're looking for safety, again, checking dosing, and then looking for early evidence of does it work and does it do what we think it's supposed to do. And then phase 3 are our largest clinical trials. The fewest number of therapies go to phase 3 clinical trials. They take the longest, but we're really looking to be sure that this thing does what it's supposed to do. And then we'll still observe for safety. Oops. All right, so I was actually really struggling with how to present this topic. So I asked my 9-year-old, and he said, "Oh, Mom, it's super easy. I can tell you in 2 seconds." And he said, and I quote, "Clinical trials are important because without them, we wouldn't have any new medicines or vaccines, and we would all be sick." He's right. I guess his mom is a clinical trialist. And so the therapies we have now and those we see in the future all have to go through clinical trials. There's oversight of the process, as we heard so beautifully explained yesterday at the FDA and other regulatory processes. There's monitoring for safety. There is extensive testing to make sure that that the drugs do what we think they're supposed to, or therapies do what they think they're supposed to. And we can even compare different therapies against each other. Okay, come on. So how are clinical trials actually conducted? And that question, this is where I struggled, could be answered in a whole lot of different ways. And so I'm just going to give you a flavor of a couple of ways that if I met you in the hall over a coffee, how I would try to explain that. It does start with an idea, right? So sometimes we observe something clinically and we say, we ought to do something about that. And here's an idea. And you'll hear from some people who had great ideas. and had to go through some trials to prove that the thing worked. There may be some off-target effects, so we're studying one thing and another thing happens, right? And then there's preclinical information that we understand from mechanisms or about how the disease process works, things we measure, biomarkers, and this is where we get those ideas, right? Whoops, these are some of our ideas, and some of them worked and some of them didn't. So again, starts with an idea up in the top left, and then you got to make a plan, right? So we write a trial protocol. Like, here's the plan for how we're going to test that thing. Then, remember I told you it's expensive to do this, so you have to get some money. Have to obtain some funding from somewhere, a sponsor, a regular, like the NIH, our good partners, philanthropy. And then there's some oversight that we have to go through, the FDA, the institutional review boards. These are all for protection of the people who are in the clinical trials and to make sure that we're doing a good job with the trial itself. Then we have to have, if we're doing a multi-site, so big clinical trials have lots of different sites that get involved, you have to pick those sites and then train them to do the protocol. So that takes time, and lots of people on those teams have to know what they're doing to do it right and do it the same way everywhere. You have to bring subjects in. So subjects are the people who actually participate and go through the trial. They get treated with whatever intervention. We measure certain outcomes. So we measure things that happen, lung function, vital signs, how you're feeling, what happens to you. You have to take that data and it's got to go somewhere and do something with it, right? And then it has to be analyzed, which is complicated and takes people with more knowledge than I have about statistics to do that, and then those results get reported out, right? That's the simple version, okay? And for every trial that gets done, this gets recapitulated, and for every drug, this gets done for every stage. So now we understand a little bit more about why that took so long and so and is so intensive. So I'm going to say a little bit more about the terminology. And you have these really nice sheets on your table that have some of the language. But this is a really simplified version of what a subject or a participant in a clinical trial might actually see. So we don't necessarily see, if we're participating in a clinical trial, all that work that goes ahead of time, all the things that happen in the lab, all of the testing, all of the writing of the protocol and the revisions and things like that, and the teams of teams that are working on this. What they see is their doctor who refers them and says, you might be a good candidate for a clinical trial. They meet a coordinator that helps them set up with an appointment. They come in and they have a screening visit or multiple screening visits. And at screening is when we look at the patient and we decide, are you a good candidate? Do you meet our inclusion and exclusion criteria? Okay, we do testing, we ask questions, and then if that person meets all the inclusion and none of the exclusion criteria, then that person can participate on the rest of the clinical trial. And they come in and they are enrolled. The process of that study starts, and maybe sometimes at that time they are allocated to what's going to happen, right? Sometimes there are different parts of the study that people go into, and sometimes that's randomly decided, so we call it randomization. Then they go through the study, and things happen to them, right? They come back for visits, they take a medicine, they do a procedure, something happens, and we follow and collect data, as I mentioned before, on what happens to them, and then the study is over. Okay, so I've got a couple of vocabulary terms that I want to go over. Your vocabulary list on your table is really beautifully done, but there are a few things there I wanted to just to add to it. So a protocol, as we said, is a written plan, the instructions for how to conduct this study. You got to have, if you're going to cook a recipe and everybody's going to cook the same cake, you got to have the same recipe. It says who, what, when, where, how, and why. Okay? The sample size, this is how many people need to be in the study that we're trying to recruit to be able to tell, it's a statistical calculation, how many people do we need to be able to tell if there's a difference between the group that's getting the intervention and the group that's being compared to? Okay, so sometimes that number is very high, sometimes that number is a bit lower. And as we're going forward to phase 3 clinical trials, that's when that sample size goes up and up and up to be really sure that the difference is real. The treatment arm is which intervention. So if there's multiple interventions during the study, there can be 1, 2, 3, 4, multiple arms of the study. But what treatment arm is what intervention that subject or group of subjects is getting assigned to based on the protocol. A control is just a comparison group. Okay, you have to be able to compare to something to know if your treatment is working, doing what you think it's going to do. And a subgroup is a smaller group of a larger group, just to double check and see, is there a different effect of what we're doing on that smaller group? Come on. So I'm not going to go through all of these levels, but understand that this is teams of teams that are taking these clinical trials on. So you have a sponsor, somebody who's running the study and taking responsibility for a lot of those aspects. There are organizations, contract research organizations in these larger studies that are there just to try to manage all of the pieces. There's a data center that coordinates all of the volumes of information that's coming in. And then there are sites. That's where you interact as a subject, as a participant with a local, a smaller group that has usually investigators, coordinators, lab technicians, and therapists and people who really gather that data and interact with the subjects. Come on. All right, so there are some challenges, not perfect. This was a very timely piece from the Atlanta Journal-Constitution, my hometown paper, about are you ready to volunteer for clinical trials? And it said, keep reading. And the author there wrote about how she had seen an article or a Facebook ad, I think, for a clinical trial and was just ruminating about her reaction to that and why she might or might not want to participate. Really interesting, her thoughts and seeing, her reaction to that. Now, I think recruitment is one of our biggest challenges with clinical trials. This is not just for pulmonary fibrosis studies, but across the board. 5% of Americans have ever participated in a clinical trial. And if you think about how many people are benefiting from clinical trials and taking medications that have been through clinical trials, that's a super small number. Why is that? Only 19% of clinical 19%, not only, percent of clinical trials are actually terminated because they can't enroll enough patients and get them to participate, according to a 2011 analysis. And 80% of clinical trials, they don't necessarily close, but they don't meet their timelines for enrollment, and so they are behind. And they don't, because of slow engagement and enrollment. Another problem with our clinical trials is diversity. And this is from therapies that were approved by the FDA in 2020. The participation in those clinical trials leading up to those approvals, 53 novel drugs were approved in 2020, and to accomplish 53 approved drugs, 32,000 patients were involved in the clinical trials leading up to this. And you can see that we have a little bit more women than men participating, far more younger patients, and far more white patients than patients of color. Come on. All right, but diversity is really important, although it adds a layer of complexity to our recruitment. We can see this because of lack of accessibility, medical mistrust, barriers in language and cultural competency. But improving this should improve our ability to provide health equity to people. Come on. Our failure rate for clinical trials is quite high. Only 10% of new therapies across the board make it through the clinical trial process to clinical utility, and I think that's actually lower in pulmonary fibrosis. Specific to pulmonary fibrosis, we have the challenges of enrichment versus exclusion, and that means that we're really trying to get a population recruited, so we're being sometimes very tight about who we let into the study, strict criteria, but the reason for that is because we're really trying to get a population that will respond to the therapy and be able to tell a difference between adding that therapy and not. In the process, however, we exclude people and we don't allow them to be a part of the clinical trials process, and in some ways we may not be targeting to the general population something that works for everybody. We may be only bringing something forward that works for a small percentage of the population. There are problems with endpoints. We use forced vital capacity primarily as our primary endpoint for studies because it does have some correlation with harder outcomes like mortality, but we have done that to the exclusion of things that impact people's day-to-day life and symptoms. With a rare disease, we have a limited study population to work with, and these trials get big, and if only a small percentage is participating, and then we have to have this number of people plus this number of controls, and then for the next study we have to have the same number of people and also a same number of people on a control arm. That adds up after a while, and there's competition for those participants in the clinical trials, and that's limited. How do we maximize that? And then we have to balance the size of the study versus the duration. And then there are some challenges now that we have background therapy and has that, the antifibrotics and things like that have potentially changed the way our disease progresses. So it's a little harder to tell a difference between a treatment arm and a control arm.
I've never been to this conference. It's wonderful to see the group of advocates and patients, as well as scientists and clinicians, all in a room together, learning together. So I thought really hard about how to give this talk, a talk on platform trials, which is a complex topic, in 15 minutes. There are different types of platform trials, and I thought that going over that generally and broadly would be less productive than really focusing specifically on one platform trial. And so that's the approach I've taken here. And I'm going to talk to you guys over the next 20 minutes about an experience we had in the I-SPY COVID platform trial. So here are my disclosures, not relevant to this talk as a consultant for direct biologics, and relevant to this talk, I did serve as co-chair of the operations of the I-SPY COVID trial and am PI of a study that's currently ongoing from the network called SPARK. So my objectives over the next 20 minutes will be to discuss the general concept of the adaptive platform trial. And then I'll start with talking about the I-SPY breast cancer platform trial, and then move in to spend most of the talk talking about the I-SPY COVID platform trial. I'm going to focus mainly on the organizational structure that we undertook and how this trial was set up. Really briefly touch on the results of that trial, and then spend 1 or 2 slides on my thoughts and summary on consideration of platform trials, both in COVID and in pulmonary fibrosis, to hopefully set up a nice discussion for the panel here. So Dr. Case set this up really nicely, and moving into her talk about clinical trials, the question is, what is an adaptive platform trial? So an adaptive platform trial allows for the testing of more than one agent or intervention for a given clinical condition. Generally, these trials involve a master protocol defining the clinical population to be studied. That master protocol has a general set of inclusion and exclusion criteria to define the population. Individual drugs or agents can be added to the protocol as study appendices instead of brand new study protocols. The interventions and agents can be dropped or added to the master protocol simply as protocol amendments, while the overall study remains open to enrollment. The sample size can be adaptive, and another key component here is that the contracts, which tend to be something that delays clinical trials getting started, can be standing and are set up with sites, and at least the I-SPY COVID approach was to not negotiate these contracts with the sponsor and the site when each drug came on the platform. They were fixed. And as opposed to a standard clinical trial where you learn all the results at the very end of the trial, the adaptive platform trial, the concept is you learn as you go. So in the traditional clinical trial, there's one protocol with one drug to be tested at a time. Completion of one agent due to full enrollment or at an interim analysis then ends the study. And then the next study starts, and there's a new protocol, a new IRB. And so this long lag time that Dr. K showed of over a decade, this is an attempt to minimize that. So this is a schematic representation of the contrast between a platform trial design and a traditional one-to-one randomized control trial design. So in the top panel, you'll see a traditional design where an individual patient is randomized either to intervention 1, or control. The patient stays on study. There may be interim analyses as the patients accumulate into the trial, and then there's a final analysis where the results are interpreted. The bottom panel here shows the schematic of a platform trial. In this schematic here, there are 2 interventions compared against 1 control group. And you can see as the study goes on, there's a new arm added, intervention number 3. And at the second interim analysis, intervention number 1 is dropped because it didn't work. And intervention 2 and intervention 3 then go on to meet the final analysis. So this slide is on the left. You'll see Dr. Laura Esserman. Dr. Esserman is a breast surgeon at UCSF. And Dr. Esserman's been at this for quite some time. And so the I-SPY COVID platform grew out of the I-SPY breast cancer platform. So the I-SPY 2 breast cancer platform has been in existence since 2010. And in this slide here, this really nicely shows the efficiency of this type of trial design. So from 2010 to 2021 here, there were 11 individual agents or combinations of agents that were tested on this platform. They were all compared to concurrent control patients that were accumulating in the trial over this decade. 7 of the agents that you'll see there in purple made it to the graduation criteria, meaning they completed the trial. And 2 of the agents there in red dropped off of the trial really early due to toxicities that were identified. And so you can see this is an open label trial. So the patient, it's not a placebo-controlled trial, it's open label. The patient and the investigator knows the drug that the patient is getting. But there are efficiencies here. There are multiple drugs on the platform being tested at the same time and compared to a control. So fast forwarding from Dr. Esserman's work over the 2010s, we found ourselves in the COVID-19 pandemic where there was a massive state of urgency for therapeutic discovery for severe disease. Okay? And we didn't know a lot as pulmonary and critical care doctors taking care of these patients in the hospital. We knew that patients that were admitted to the hospital with severe COVID-19 were at risk of mortality, but not everyone died. In some ways that was similar to cancer. And we thought that creating a platform trial and creating a phase 2 trial in particular might be important to think about discovery of new agents. And we thought that this should be independent of the vaccine efforts, which were massive and obviously tremendously successful. And on the right-hand side, you can see, as we all recall, in 2021, the leading cause of death being COVID-19 in the United States that went on for quite some time. So there were a few assumptions that we made and decisions as we went into this period thinking about how to design this trial. One was that as an ARDS researcher over the past decade, we'd come to appreciate that ARDS was increasingly a heterogeneous syndrome. But we thought maybe with a single etiology, the SARS-CoV-2 virus, causing severe pneumonia and ARDS, that we would have a reduced heterogeneity of this syndrome. We thought that a drug with a big clinical effect would be needed to save lives during the pandemic. Another concept that we really wrestled with at the beginning of this and that we changed slightly from the I-SPY breast cancer design was that consenting a patient or a surrogate in the urgency and crisis of an inpatient COVID-19 hospitalization to up to 4 possible drugs was too complex. And we made a decision up front that we would generally approach patients and families about interest in a clinical trial. And if they agreed to participate, we would randomize the patients first and then approach the patient or surrogate with a consent for that individual agent. We thought that concurrent controls, meaning controls that were matched contemporaneously during time of the pandemic, would be important because we knew and anticipated that the standards of care of treating patients with severe COVID would change over time, which it did. We thought really long and hard about placebos and whether we would want a placebo in this trial, but we eventually discarded that idea because we thought that it would increase expense and slow down our ability to move agents through this trial rapidly. For instance, of the first drugs on the platform, 3 of them had different routes of administration. So one was oral, one was subcutaneous, and one was intravenous. Pretty hard to get placebos for all of those. So here's a slide on the I-SPY COVID study structure. The study, as I mentioned, was funded by a not-for-profit out of the Bay Area, Quantum Leap Healthcare Collaborative. They also served as the data coordinating center. The PIs of the study, Dr. Esserman, Calfee, and Liu. The funding actually was quite interesting and came from a mix of industry sponsors as well as federal funding to support this initiative. Some of the committees, there was a massive amount of work that went into this trial, and some of the committees are listed below. The names are not important, but just to show, as Dr. Case illustrated, that these trials are complex and there were multiple committees working in parallel to pull off this work. So this is a little bit of detail about the statistical design, and this was informed by some of those assumptions that we had going into this. So we decided on a Bayesian analytical framework with an adaptive sample size. We thought that we would enroll anywhere from 40 to 125 patients per arm. We had a data monitoring committee that was really robust and amazing and met every 2 weeks to evaluate the data coming out of this trial. And individual agents, 2 things could happen to individual agents on this trial. They could either graduate, meaning that there was likely benefit, and the rules for graduation in this trial were based on a Bayesian framework of a posterior probability of a hazard ratio for recovery, which was defined in this trial as ability to reduce the amount of oxygen that the patient was on below their baseline entry for 48 hours, needed to be greater than 97%, or that the drug was reducing mortality with a hazard ratio less than 1, with a posterior probability greater than 90%. To meet the criteria for futility, you needed a posterior probability hazard ratio of recovery less than 1.5, needed to be greater than 90%, and a hazard ratio for mortality less than 1 being less than 50. So that's a lot of statistical jargon, but we set a high bar to get a study through this trial, and we were willing to discard drugs that were unlikely to help very quickly. And so we acknowledged there was a risk of a type 2 error with this approach, meaning that maybe some of these drugs would have shown a smaller effect, but we didn't care about that when we designed this trial. Okay, so the top slide looks is a schematic of what the study looked like. So we screened hospitalized patients with COVID. They were on greater than 6 liters of nasal oxygen for participation in the study. They were approached by the individual patients or their surrogate, randomized to one of a number of active agents on the platform. We had up to 4 active agents on the platform at a single time. And the primary outcomes are listed on the right. You'll see there that time to recovery was originally listed as our primary outcome about midway through the trial due to the data monitoring committee. We changed the primary outcome to be a combined outcome of time to recovery and mortality. So here's our network that was put together extremely rapidly and was primarily using sites that were in the I-SPY breast cancer network. So we had a really wide geographic distribution. We have academic and community hospitals that are part of this network. And when we were operating at our maximal enrollment, we had 42 trial sites. We enrolled over 3,000 patients. And we evaluated 12 agents and completed 11 study arms in 1.5 years. Just a drastic change in that decade timeline of getting a drug through testing. So we had a rigorous process for agent selection. The first 4 drugs on the platform were selected by something called the COVID R&D Consortium. This is a consortium of industry partners that were willing to donate their drugs onto this platform. Those are the 4 drugs: apremilast, a PDE4 inhibitor, cenicriviroc, which is a drug that has antifibrotic properties, and antiviral properties, razuprotafib, which is a Tie2 activator, and icatibant, which is a drug used for angioedema that blocks bradykinin release. And subsequent drugs that were selected were presented by either internal or external scientists that could come in and present to the I-SPY COVID Agent Selection Committee. There were other efficiencies of this trial, I think, which are critical to pulling it off. One is a centralized IRB, which is really the standard for most clinical trials now, but I will make a point to say you need an IRB that is, has experience with adaptive platform trials because it's a different beast compared to a traditional trial. We had a centralized drug supply and distribution. The Medicare coverage analysis was sort of standardized and was distributed with new agents being added. And about midway through this trial, about half of the sites implemented an automated electronic medical record data extraction platform that pulled data automatically into the electronic, into the case report form through the site EMR. So this is the publication that that was published a few months ago in Lancet eClinical Medicine. The details of this are really not super important for the group here, but needless to say, those of you who are interested or who often look at clinical trials in journals, the consort flowchart here looks a bit different, and that is as we come down from the number of patients screened to the patient's number allocated in these different arms, we have 7 arms at the bottom instead of 1 single arm versus a placebo. So we decided to lump all of this information from the first 7 agents into this report. None of these first 7 drugs met the criteria for graduation. And these are the primary endpoints of the drugs, these 7 drugs tested on the platform. There's 6 shown here. One of the drugs was dropped off of the platform due to feasibility issues. But I'll point out a couple of things here. This is the time to recovery, one of the co-primary endpoints here. The active drug is in the colored bar, and the control is in the gray bar. You can see that there's no difference in the time to recovery. One of the drugs here that you'll see in panel E, a combination famotidine and celecoxib, which had a very small study with historical controls used, actually showed significantly worsening time to recovery. And you can see that again, in panel E with that drug, looking at mortality here, that drug fared worse both in mortality and in time to recovery, and was efficiently moved off the platform after 40 patients were enrolled. So this is the schematic of the drugs tested on the platform. We had up to 4 at a single time, and you can see how the different drugs cycled through this over that period of 1.5 years. The last drug that was added to the platform, imatinib, a tyrosine kinase inhibitor, was added, but the pandemic slowed and is currently on pause. There were other real serious challenges in conducting open-label critical care trials during the COVID pandemic, one of them being how to adjudicate and measure adverse events. How do we determine these drugs are safe? Given the complex environment of the ICU where adverse events occur every day, multiple times per day, this is complicated as well by this open-label design. And one of the innovative things I think that we designed in this trial was to create a case report form that the study investigators filled out every single day that a patient was on a study, whether they were on an active drug or on a concurrent control. We measured clinically important things to us in the ICU, such as, did the patient develop a deep venous thrombosis? Did they develop a pulmonary embolism? Did they have a myocardial infarction? Did they go on dialysis? We systematically recorded all of this information, and every 2 weeks this was evaluated by the Data and Monitoring Committee. So in early 2023, enrollment in the interventional arm of ICE-by-COVID paused, as I mentioned, and we rapidly, without stopping the protocol, we added a new appendix and transitioned this platform to a biomarker-based observational study to survey the landscape and prepare for a ARDS trial that was agnostic to your COVID-19 status. And this is the sub-study that we're currently conducting. Those are the participating sites. We call this the SPARK trial, subphenotypes of ARDS using rapid prospective collection. The goal is to assess the feasibility of enrollment and assessment of patients with acute hypoxic respiratory failure and ARDS into subphenotypes to inform a future investigational trial. This work a lot of this was based on Carolyn Calfee at UCSF's work showing that patients with ARDS can be classified into hyper and hypoinflammatory phenotypes with a combination of clinical data and biomarkers, and we are employing a couple of rapid point-of-care biomarker tests that we are incorporating into the study design. So this is some of the data coming out of this trial. As of last week, we had enrolled 116 patients with a nice mixture of patients with acute hypoxic respiratory failure and ARDS. We're able to phenotype these patients biologically about 1.7 hours from the time that blood is obtained to go in this biomarker assay. And you can see in the bottom right that we have about 19% are hyperinflammatory and 80% hypoinflammatory. And so we're going to use this data to inform the next trial that we're going to launch in this network. So here's some lessons learned from the ICE-by-COVID experience. A platform design is an efficient way to evaluate multiple agents for a clinical condition rapidly. There's some compromises that are made with the master protocol and agent appendices that can lead to a series of issues that can create group imbalances that may be worth the risk. We were optimistic that COVID ARDS would be less heterogeneous, but we were wrong. We were overly ambitious about a big clinical effect, and our design may have led to some type 2 error that we discarded agents that actually may be helpful. And some of the drugs that we tested on this platform, I think, should be tested further. The early futility rules, which I mentioned, were especially useful in the wild pandemic environment, and we have a good example of how this trial eliminated a drug combination quickly. The industry sponsor and academic partnership that I think grew out of the ICE-by breast cancer experience was really really interesting, and it offers really significant advantages, and I think can be optimized. We thought that limiting, and as we move forward, we would limit the active drugs to 2 to 3 on a platform, and we likely needed a larger sample size. And lastly, we think that the addition of biomarkers or predictive enrichment, which I know the ILD field is very good at this kind of work, is an optimal way to think about an adaptive platform trial design. So here's the funding statement. There's my email at the end. And again, I'd like to say thank you for inviting me and look forward to the discussion. Thank you.
Good morning, everyone, and thank you to the PFF for the invitation to speak with you all this morning. I was struck as I walked in yesterday evening to dinner, as I am every time I attend the PFF Summit, and witnessed the conversations that were occurring across dinner amongst all of us, at how unique and powerful and logical it is to bring broad stakeholders to a conference and to facilitate crosstalk while centering the experience of patients. That is a catalyst for change, and one that is really unique to this summit. And my goal today is really to introduce or talk about how AI can help us in the mission-driven work that brings us all here. The talk is really predicated on the assumption that AI has a role in medicine and therefore a role in clinical trials. And maybe you are in enthusiastic agreement with me on that. Or maybe that statement lands with you with a hefty dose of skepticism. But either way, I think that we can all recognize that AI has really asserted itself into our lives, whether we like it or not, right? And the intensity with which it has done that can make us really feel like we are passive bystanders to the process. And I want to challenge that notion for all of you, because the same expertise and engagement that you bring to this conference is needed in discussions about how, when, and why AI is used in both our clinical and research workflows. And so I'm going to talk about some ways that it is showing up in our clinical workflows and specifically within clinical trials today. So why are we talking about it in clinical trials? Well, we've heard from the last two talks that there are major barriers to the way we traditionally run clinical trials, right? They are time, labor, and cost intensive, and only a fraction of patients who could be eligible for participation in a clinical trial are actually engaged. And one of the common themes to those barriers is the immense amount of data that is needed to run a clinical trial, right? You have to collect the data, store it, manage it, analyze it, and then redistribute it. And that requires an intense amount of resources and man hours. And one potential way to leverage AI is to help to offload some of that work and automate it. And so this is an outline of the different ways in which AI can be integrated into clinical trials. And this is not meant to be exhaustive, but it gives us a sense of different, an overview about different ways that it can be used. And I'm going to try to walk through some of these examples. But before I do that, I want to make sure that we have a common set of terms with which to approach this topic. So to begin with, data science is a really big overarching term that really refers to a field of study where the goal is to extract meaningful insights from data. Artificial intelligence falls within that field, and it goes further with the goal of enabling computers to mimic human intelligence by programming in some logic and then allowing the computer to apply that logic to a new set of data. Machine learning then goes a step further and allows the machine to actually improve at the task that it's been given through experience. So with each iteration of doing that task, it learns from its mistakes and improves its ability to perform the task without that learning being explicitly programmed by humans. The machine is sort of learning on its own. And then deep learning goes a step further and sort of like cracks open the amount of data that the machine can bring in. So now we're talking about massive amounts of data that are used for the input, allowing it to quickly process that data and then map that input to an output. Okay, so with those terms in mind, I want to start by dispelling a common myth when we talk about technology and AI, and that's that you can take someone who knows how to program, take a large data set, and all of a sudden it's going to be eureka, and we're going to have all of this new scientific knowledge and understanding. And that is untrue because, at least for now, humans and machines learn fundamentally differently. Humans learn by taking a limited number of observations and being able to glean meaning from that because we can layer in experience and a broader context and assign biological plausibility. I have not seen 100,000 CT scans, but I can diagnose a patient with pulmonary fibrosis pretty well because of my clinical training, the patients I've seen in my experience, conferences like this, all of that sort of information I can layer in and bring to a single encounter and inform the data that I'm seeing from that one patient. Computers don't learn that way. Computers learn by just sort of taking in lots and lots of data and then finding patterns in that data that may explain its variability. But absent any context or biological plausibility, a computer is just as likely to generate nonsense as it is to generate really new insights. Or worse, even harmful mistruths, because we know that the data sets that our machines are trained off of are flawed. And so really what I'm talking about when I say artificial intelligence is augmented intelligence. And that's taking human intelligence, where we bring domain expertise, and combine it with the computing power of machines, and that combination is a pathway for scientific knowledge. Okay, so now let's talk about, I was doing so well. Okay, let's talk about some of these algorithms in action. And I'm gonna start with patient identification because that is the area where I think there's the most applied examples in clinical trials, including in ILD and pulmonary fibrosis. So we've heard that patient identification is really hard. If you are connected to a care center network, then you are being seen at a center that has a framework for identifying patients with pulmonary fibrosis. But we know that's not true for many or most patients. And so if we want to think about diversifying where we are recruiting patients for, that requires going to health systems that currently don't have a clue how many patients they have in their health system that has pulmonary fibrosis. So how are we going to do that? Well, one way is to automate the identification. And one of the first steps in trying to automate was saying, well, can we take codes? Physicians see patients and they put in these diagnostic codes. Can we take those diagnostic codes? Can we combine them with some demographics like an age cutoff and some signal that the patient has gone through a diagnostic evaluation like a CT scan and create a signal that a patient may have idiopathic pulmonary fibrosis? And then we iterated on that and said, well, can we not just focus on the variables that we know that are important, but patients move through the health system and generate tons of information in doing so. There are thousands of variables available in an electronic health record. Can we harness all of those variables and create a more complex signal that allows us to more accurately identify patients with interstitial lung disease? And so this will be diagnostic codes, comorbidities, lab data, and going into clinical notes and using natural language processing to extract meaning. And in doing that, we were able to not just generate one model, but multiple models that could be adjusted based on what variables were in any given data set, again with the goal of trying to automate the identification of patients. Another approach to doing this was even prior to the diagnosis of pulmonary fibrosis, patients are seen in a health system. We know the sort of increase in frequency with which patients are seen starts almost 5 years before the diagnosis. And they get diagnosed with all different types of comorbidities. Is there a comorbidity signature that we can use to identify patients who are at risk of developing pulmonary fibrosis? And again, with each of these iterations and increasing the amount of variables that we are feeding to the machine, we are getting better and better at identifying patients automatically who have pulmonary fibrosis. And then the other approach to doing this is to say, let's stop centering diagnostic codes, because as clinicians, we are terrible at putting those in. Can we just maybe really look at the images? And can we teach the machine to look at an image and be able to say, this patient has hypersensitivity pneumonitis, this patient has IPF, because of all of the complex data that's captured in a CT scan? It's exciting for me to see that this type of work, this type of algorithmic patient identification work is happening not just broadly in medicine, but also in pulmonary fibrosis. And we now have a number of different algorithms that can be used to identify pulmonary fibrosis and subtypes. So what's the use of this in a clinical trial? Well, the goal would be to be able to run one of these algorithms in a data set, say give it to a health system, to be able to pre-screen how many patients would be potentially eligible for a clinical trial. So you could start by running an algorithm to identify progressive pulmonary fibrosis, and then you can layer in inclusion and exclusion criteria for a specific trial. And then all of a sudden, a health system or an investigator can have a really clear understanding, can quantify how many patients would be potentially eligible for a clinical trial and make data-driven, informed decisions about whether a specific trial is a good fit for their patient population. Ideally, this could also be run in real time. So as patients are being seen in the clinic, we are able to identify new patients as they go through their journey with pulmonary fibrosis who become newly eligible for a clinical trial. And maybe not just newly eligible for one, but now maybe we're able to identify if a given patient in front of us is eligible for multiple clinical trials. And then the conversation is very different. Not can I offer you a clinical trial, but what clinical trial is best suited for you now that I know what your options are. And this is something that our colleagues in oncology have been really, really good at. Trial design. This area, I think, is exploding, and I could give an entire talk just on how AI can inform trial design, but I just want to emphasize 3 areas. The first is that algorithms can be used to evaluate prior clinical trials, phase 1 and phase 2 studies, and then use that information to inform the design of phase 2 and phase 3 trials. That includes being able to simulate how changes in the inclusion and exclusion criteria will impact endpoints, enrollment targets, et cetera. The second is it can be used to help leverage historical control. So we've heard a lot about how finding control populations for placebo can be really hard, particularly as our numbers increase and trials continue over time. And so one goal has always been, well, can we leverage the data from historical controls and use that for a current trial? And the biggest challenge there is really feeling confident that historical controls match the population of our current trial. AI algorithms can help really interrogate that matching and make us feel more confident that the historical control data is relevant to our current questions at hand. And then the third is to help innovate how we design trials themselves. And we've heard a lot about adaptive trials. Adaptive trials require interim analysis and really being able to evaluate and then adjust the study time and learning as we go. Learning as we go requires evaluating more data, and AI algorithms can help in that evaluation process. An example of this that's happening in ILD is the REMAP ILD adaptive trial platform, and I believe there's a session later today that's going to talk in more detail about how we are applying the concepts of an adaptive trial platform to further research in interstitial lung disease. And then another trial design innovation is really thinking about decentralized or virtual trials. So now the idea here is to take away our standard idea that we have trial centers at our hospital centers where patients have to come to us for a clinical trial, and instead we're bringing clinical trials into the homes of patients. That is that allows us to kind of open up who can be involved in a clinical trial, but it also makes us need to think about innovative ways that we're going to collect data, because now a lot of our sort of monitoring is going to need to be done in a different way. And doing monitoring in a different way means that we're going to be collecting new data streams that require innovative ways to analyze and manage that data, unless we want to drive clinicians and investigators crazy. And so AI algorithms can help do that. Data management. So I think I've sort of made this point already. But over the course of a life cycle of a clinical trial, there are multiple stages of collecting and analyzing data. From our initial stages of collecting the data, preparing it, storing it, analyzing it, and then distributing it back to clinicians, to investigators, and to participants. Machines are infinitely faster than humans. I mean, this sort of estimate of them being 125,000 times faster is already outdated by the time I've made this slide. They are much better at data processing. So how can we leverage that? Well, we can leverage it by running quality control algorithms in the background to improve the integrity of the data. When we're collecting data from multiple different sites, there's challenges with harmonizing that data because it's always collected a little bit differently. Algorithms can help in that process. They can help with interim analysis, and they can help with allowing us to collect novel data streams, whether that be remote monitoring data, information from electronic PROs, nutrition and exercise data, or also allowing us to integrate data from other data sets that really inform and expand our understanding of pulmonary fibrosis, including genetic and environmental data. Recruitment. I think Dr. Case made the point that recruitment is probably the biggest challenge in clinical trials. And so one way to do that is to reach outside of our traditional methods for recruiting patients and thinking about how we can leverage social media to reach out, do direct-to-patient advertising about clinical trials. In the same way that your feeds on your Twitter profile are curated, and the advertisements that you see about, the new shoes that are out there are curated, we can also curate the content about clinical trials and push that out to patients and caregivers where it may be relevant. And then it's not just enough to recruit patients, but we need to retain them in clinical trials. And one challenge in retention is the ability to communicate with participants during the clinical trial. And we have seen efforts to really use AI to improve and enhance communication and patient experience, both in our clinical fields, but also in research. And so large language models, generative AI, which can provide some early communication, bidirectional communication with patients, is one way to identify patients who may need additional support while participating in a clinical trial. And then the last point is compliance. And this is a long list, and it sort of doesn't even scratch the surface of how much work needs to be done during a clinical trial for compliance. And the example that I will really emphasize is monitoring for adverse events. Our cardiology colleagues have done studies that have compared traditional approaches to monitoring to adverse events versus AI models that can monitor for adverse events, and have shown them to be equivalent. And then beyond that, AI algorithms can actually see patients who are at risk for an adverse event before it happens, allowing for earlier intervention and maybe even avoiding that adverse event. And so I hope that I have shown you sort of the potential and power of bringing AI into clinical trials. And so what are the concerns? Well, there are many, and I'm just going to highlight three. The first is that these algorithms, the more data we put into them, the better they perform. But the less able we are to explain them, to understand them, or really to interrogate what's driving them. And so there is a tension between the performance of an algorithm and trust and understanding amongst potential users. There's data quality. The algorithms are running on flawed data sets. And so fundamentally, we need better data sets. And if that's something you are interested in, please come and find me afterwards, because that is a nut I am trying to crack. But in the meantime, I think it is important that we recognize the bias that exists in the data sets that we are giving algorithms and training them on, and being really thoughtful that in deploying algorithms, we are not compounding the impact of bias on our patient populations and decision making. And then last is data privacy and security. As we amass these large data sets, we have a responsibility to think about also putting in the appropriate measures to make sure that that data is safe and secure and respects the trust that patients have in providing their data. And that's it.
Thanks to the PFF for inviting me to give this talk, and the title is Emerging Devices Used in Clinical Trials, Device Observational and Implementation. This was a slide provided by the PFF. It's a lot of writing, but basically I don't have any relevant disclosures or conflicts of interest related to this talk. And I should preface, this is where I guess imposter syndrome comes in. I am not necessarily an expert on these novel devices or implementing them in clinical trials, but I think it gives me a good perspective of reviewing the data that's out there, talking to the actual experts, and kind of putting it together for this audience. And so the overview of this talk is three major parts. One is, what is the rationale for trying to design and implement these new devices and incorporating them into clinical trials? Part two is potential candidates, and there's three major ones listed here, and then we'll touch on potential barriers of how we implement these devices into clinical trials. So as Dr. Fran mentioned, there's a real push for these decentralized clinical trials, and it was really accelerated by the pandemic. And as Dr. Fran mentioned, these decentralized clinical trials, what the concept is taking the elements of a clinical trial, so whether that's drug delivery, adverse event monitoring, or clinical trial endpoint adjudication or capturing it, taking that, which is typically done at the clinical centers, at the study sites, but bringing it to the patient's home. And there's a potential advantages of this approach. And so one is actually increasing the sample size. It would probably cut down a lot in terms of resources and time of being able to screen and recruit patients into clinical trials. Two is actually decreasing the time. So I think our study coordinators, our nurses, our site investigators, and particularly our patients and families can appreciate how long these study visits can take when you're enrolled in a clinical trial. If we're able to integrate these devices and do remote monitoring, it may cut down time, it may cut down on the travel time to get to the study site to do these trials. So this is a potential advantage. And then another major potential advantage is actually attenuating potential inequity that our speakers have kind of touched on about these clinical trials. And so case in point, a lot of our clinical centers that are participating in clinical trials, they tend to be in suburban and urban centers. And so we can think of patients in more rural settings or hours away from these study centers not being recruited or unable to be recruited to these clinical trials. If we have this ability to at least remotely monitor, this may get at that. Sorry, it's very sensitive. So there's other diseases that have actually been doing this for quite some time, and particularly diabetes and cardiovascular disease. And so they have been able to design, implement devices that can monitor clinically in terms of disease progression, such as continuous glucose monitoring. But even as of recent, they've been able to implement these devices as part of the clinical trial and capturing the endpoint. And so what would make an ideal clinical trial endpoint device? And so one is it has to reliably capture disease progression. And an example is we typically think of a surrogate marker of disease progression as the change in FEVC over time. Well, if we're able to try to capture that at home, can we really put trust that it reliably captures it as if we were to do it at a clinical site getting spirometry by a trained respiratory technician? Is it comparable to sort of standard of care? The device ideally has to be very easy to use by the subject at their home and is a minimal burden. And then the device itself ideally should not exclude potential subjects from being enrolled in a clinical trial if it's dependent on reliable wireless internet, it's dependent on certain quality of smartphones or tablets, again, it may exclude certain populations. And then I think most importantly, it has to be a device that minimizes the reliance on a subject's effort and coordination in using the device. And so probably the most well-studied of these potential devices to incorporate into clinical trials is home spirometry. And so these are examples of home spirometry devices, and typically it's handheld, and it's usually paired with some sort of smartphone or tablet or computer. And then the patient breathes in, and then the spirometry captures the measurement, and it gets delivered to the smartphone, and it gets uploaded to some sort of web-based or cloud system. And you're able to have this uploaded, and your provider can potentially monitor your FEVC measurements. It can follow it over time. And if there's a big dip or something changes, potentially they can incorporate that and reach out to you and see if anything has changed. And there is FDA approval for some of these devices for clinical monitoring. In terms of its use in clinical trials, the jury is still largely out. Oop. And so this is a figure from this really nice review by Dr. Toby Mayer out at USC, where they kind of reviewed sort of these devices, and particularly home spirometry. And so these are correlation plots. And basically what they looked at was how well do home spirometry measurements, specifically FEVC, how well does it correlate to an on-site clinical spirometry measurement? And so strong correlation here is indicated by these dots that represent each patient. And you want those to be grouped together, forming a line at a 45-degree angle. And if that's the case, you have a line going through those points, that's good correlation. And so at least at a single time point at baseline, it's pretty well correlated. So home spirometry FEVC was very well correlated with on-site spirometry FEVC. But when you look at changes of this FEVC over time from home spirometry, the correlation is significantly weakened. And we know this is important because typically, again, we use changes in FEVC over time as one of our clinical trial endpoints commonly. And so you can see here on these correlation plots from several different studies that have looked at home spirometry, the dots are a bit scattered, the lines are not at that nice 45-degree angle, they're kind of skewed. And so we're not able to really confidently say whether home spirometry FEVC over time really mirrors what you would get in the clinic. And so some other considerations to take with home spirometry is, again, it relies on human effort. And this is a question that's ongoing, I think, with providers, clinicians, and researchers is like, how many maneuvers? And so some of the studies you look at, they had patients do a home spirometry 3 times a week. Some of them had them do 3 times a day, once a week. And it's unclear what is the ideal number of measurements of home spirometry to be done that can reliably capture disease progression. In those studies, they also comment that there were very extreme values of FEVC, very high, very low compared to prior or subsequent FEVCs. And so whether that's a real measurement or whether that's artifact, and how do these algorithms that sort of account for all this data, how do you account for these extreme values? And then more of an existential question is, what is a true FEVC? We've always assumed and relied on on-site spirometry, but possibly is it home spirometry, your measurements at home over time and a lot more data, is that actually your true FEVC? And I think one of the most important issues and barriers in terms of trying to put home spirometry in a clinical trial is actually the adherence. And so this is a figure from a paper from the INMARK study that actually had a component of home spirometry. And you can see here that over 80% of patients actually were somewhat adherent over time. But then I think you can sort of eyeball and see that the adherence does decrease over time in this clinical trial. It was about 52 weeks. And if you talk to clinicians and researchers that have tried to incorporate home spirometry, it's usually about 6 months is when it wanes, the adherence. And so if you're going to use home spirometry as your primary endpoint, you got to have people use it, right? So what are the ways we can at least improve adherence and make it easier for patients to use this regularly? And then they also noted that patients who had less disease severity tended to have better adherence. And so it's trying to untangle whether it's because patients who are sicker or getting worse in your clinical trial, they just don't have the energy or the strength to be able to complete these measurements as frequent. The next device I'll talk about is pulse oximeter, and I would point to this really nice perspective piece by Carrie Anderson, Susan Jacobs, Don Rapola, and Jess Swirgus. And it's a nice perspective, and they kind of discuss whether we need to think about incorporating oxygen need as a clinical trial endpoint. And so what they point out was that oxygen needs is a very much patient-centered outcome, and the development of a new or increased need of oxygen definitely indicates a new stage of life for these patients, their family, and it's probably one of the strongest markers we have of disease progression. And in their perspective piece, they mention about how to reliably capture oxygen. And I do think that we probably need to look beyond our standard approach of measuring oxygen levels and developing new oximeter devices. And so some of the features that we need to think about as very smart people and companies are working on this is we probably want a continuous oxygen monitor. And the key point is that it minimally interferes with daily activities for the patient. And so again, you could think of it from other diseases such as continuous glucose monitoring, which has probably transformed patient care and clinical trial design for diabetes. Can we design and implement something similar? And I think something that's very nuanced is whether we can design oximeters that can adjust for certain environmental factors that can influence one's oxygen level. And so we know things like altitude, air quality, barometric pressure, humidity, people's activities, whether they're exercising or doing something more strenuous. Can we design devices that can account for this? And then it's not just relying on the oxygen level by itself, but actually integrating it with what they're doing, how they're feeling, symptom logs, cough, were they stressed about something at home, were they exercising again, were they grocery shopping, were they doing heavy lifting, integrating that with these oxygen levels and making this a much more nuanced and integrated approach of how we incorporate oxygen into these clinical trials. And then lastly, as many of you are aware of, just recent research, data, and obviously a huge priority at the FDA is how do we account for skin pigmentation in these sort of newer, more novel oxygen devices. And this is an example here of a potential way of how we can think about these novel oxygen devices is a device that pretty much looks like an AirPod that's on the ear that you can clearly envision a patient having this and has minimal impairment on their daily activities. And so then the last potential candidate I'll talk about is blood-based biomarkers. And so I think as many of you are aware in this room, it feels like at every PFF Summit, every conference, there's just always been this interest of developing and validating blood-based biomarkers to both ensure clinical trial enrichment and also to assess treatment response. And I think at least the INMARK study demonstrated feasibility. So this was a study where the primary clinical trial endpoint was extracellular matrix neoepitopes in the blood. And although it wasn't a positive study, they actually showed at least it was feasible to collect this blood over time and use it as a primary outcome. In the last few years, and as other people have talked about at this conference, there's been a lot of advances in semi-quantitative proteomic platforms that have enabled us and other researchers to identify potential novel candidate biomarkers, both for clinical trial enrichment and to track treatment response. And this is a figure from Dr. Oldham's recent paper in the Blue Journal out of Michigan, where we see here that he derived a proteomic score of several hundred proteins, and the red line there indicates those patients who had a much higher proteomic score, and you can see that they had much shorter survival. And I think what makes this very interesting is that's probably the group of patients that we want to quickly enroll in clinical trials and monitor them. Now, unfortunately, with with blood-based biomarkers, currently it relies on in-person lab draws. So you can already start to think of the dread of having a blood-based biomarker as your clinical trial endpoint. You're probably wanting to follow it over a period of time. it's asking us a lot for these patients to come in every 4 weeks, 6 weeks, and getting a blood draw during that time. And so just something to be mindful of. So this would be my one outrageous sort of pie-in-the-sky idea as we start to continue to work on identifying these blood-based biomarkers. So I would say it's not just identifying and validating these biomarkers in the blood for lab draws, but whether we can take a further step and do dry blood spot. And so it's a well-established approach. I think all of you are very familiar with finger pricks. It's a very simple procedure, and it would allow for remote sample collection. So it gets back to this concept of decentralized clinical trial. And so what we can imagine is the research team mails this finger stick kit to the patient at their home. Patient drops the blood on the card, sends it back, mails it back, and that's how we can potentially measure these biomarkers. And notably, these dry blood spots are actually stable at room temperature for years to decades. So it gets at these issues with processing and storage with in-lab blood draws. And so this is a slide courtesy of Dr. Monica Parker and Dr. Linda Steyer out of the New York State Department of Health. And this is an example of what we can maybe envision in the future of how we incorporate these biomarkers in at least pulmonary fibrosis clinical trials. And so here, this is an example. You have a card with these dry blood spots, and all it takes is like a punch hole sample. So you do a punch hole, literally, and then you run it in these immunosorbent assays. And the New York State Department of Health used this actually during the pandemic to sort of track antibody levels. And here, this is a correlation plot. And what they did was they correlated the levels of these COVID antibodies from the dry blood spot with serum levels from the lab. And you can see again here, those dots are mostly together forming that 45-degree line. And what they found was at least the dry blood spot measurements of antibody levels strongly correlated with the in-lab measurements. And there's other research showing that other biomarkers like D-dimer and interleukin-6 are strongly correlated between dry blood spot and in-lab. So I would challenge us as researchers, investigators, to not just think about in-lab blood draws, but potentially looking about dry blood spot. Sorry. So almost done, but other considerations to take, again, is back to this home devices. So again, relying on smartphones, tablets. And I think something to be mindful of are these software updates to devices. You know, whether they reset the settings, they reset the device, they reset the app. These are things we have to factor in if we're incorporating this into a clinical trial. And then one of the sort of nuanced issues is blinding. So we do know that patients who have access to their measurements, who see what their FEVC is, they tend to continue to use the spirometer. If we were to blind them in a clinical trial, you know, it probably would have an impact on adherence. So how do we reconcile that with study design? And then reiterating that we probably need to integrate all of these potential endpoints captured by these novel devices together. And I didn't get to it, but we can also foresee how this might be useful for medical monitoring and identifying adverse events by the research team. So in summary, again, to reiterate, there's a crucial need to implement decentralized clinical trials for pulmonary fibrosis. It will be very dependent on these novel devices to capture clinical trial endpoints and for monitoring. But I would say there's a lot of work that still needs to be done to validate, to see whether it really captures disease progression. And I think most of all, to design these devices that minimize the burden on patients, clinical coordinators, nurses, and the physicians and investigators. And then I just want to end on this note. I found this picture, actually. So this was the first PFF Summit I went to in 2017, and there's probably some familiar faces here. And I guess that's like 6 years since then, and it's been amazing seeing the remarkable progress being done in this field with the research, with the patient involvement in the clinical trials. And so I'm very hopeful. I'm very excited where we're at right now, where we're definitely, I think, becoming more nuanced in how we approach these clinical trials, of making it much more patient-centered and personalized. And so I'm excited for the next coming years with all this data and research that's coming. I just want to thank all the people here, and particularly Toby Mayer at USC, who kindly looked over my slides because he's the real expert. So thanks, everybody.
I am a IPF patient, originally diagnosed in 2017. I say originally because, like a common story for most PF patients, we tend to go through a diagnosis and a misdiagnosis. So I have no disclosures. I'm retired. So I'm a visual type person. I like to share my stories in pictures. So way back in 2017, I had a nagging cough. I thought it was just a influenza that was left over from the holiday period. Ended up going to my local GP. He thought it was pneumonia. After a 2-week course of antibiotics, I'm back at the GP. He gives me a referral to a local community pulmonologist, and he gives me an x-ray, really not sure, sends me back for a high-resolution CAT scan, and comes back with probable pulmonary fibrosis, and I'd never heard this word or this term before, so kind of common journey that most PF patients go along. Dr. Google come up with the 3 to 5 years that people have talked about the last couple of days. And then so I've got pneumonia, I've got pulmonary fibrosis. 2018, and I immediately start on one of the anti-fibrotic drugs because the potential pulmonary fibrosis. Early 2018, I'm at a research hospital close to where I lived in San Diego, and my pulmonologist is going over the results of my second HRCT scan and says, you know, you probably need to prepare yourself. This is progressing. You probably need to prepare yourself for a transplant within 12 months. And that was a change in life. That one discussion with one sentence changed our whole lives because, you know, now we're planning retirement. I'm thinking 12 months. I do the research on the data for lung transplantation. Only 2% of pulmonary fibrosis patients get transplanted. So those are not odds I would take to Vegas. I'm thinking that's never going to happen to me. So he said, well, if you really need, and then he starts talking about hypersensitivity pneumonitis. So now he kind of opened this door of uncertainty on what my diagnosis was again. So, and said to me, if you really want a definitive diagnosis, the only way at the moment is really to go for an open lung biopsy. So I make an appointment with the lung surgeon. He points out, he uses the end of his thumb, and he says, I'm going to take 3 tissue samples this size from your diseased lung. And suddenly I thought, no, you're not. I go home, I do the research, I look at the mortality rates of open lung biopsies. 16% was that number for both elective and non-elective, but elective being a lot less than 16%, but still a pretty scary number. So I had spent 4 years in genomics. I was working for a company that was making next-generation sequencing equipment and all the reagents for doing sequencing and DNA analysis. So I'd seen all the information that was being published on NIPT at the time. I was looking at the cancer biomarkers, and I thought, there has to be something different. There has to be something different than punching 3 thumb-sized holes in my already diseased lung. So it kind of set me off on a journey. I came across a customer of the company I was working for, and they were working on a test, and we heard some of these words this morning, but it was a genomic classifier using machine learning and artificial intelligence, and I thought, this is it. This is what I need. I reached out to the company. Immediately on the portal, I got immediate contact back saying, hey, appreciate your reaching out to us. However, we're still in clinical trials. And that was kind of my first thought about clinical trials within this journey. So they said, when we have received approval, we'll come back out to you. So, and that's what happened. So 2 months later, I get a phone call back from the same company. They said, hey, we have this. It's been FDA approved. You would qualify. And it's available at UCLA. So I transitioned from one research hospital to UCLA. I go through the test. I comes back with a positive UIP pattern, not the result I wanted, but now it was enough information that I could begin, you know, my treatment for the future, knowing that I have a definitive test for IPF. So when we get asked about, you know, why we as patients want to look at why a patient wants to participate, just like Amy's 9-year-old son, we want to stay alive. We don't want to get sick. So I have this analogy where we talk about without patients, there are no clinical trials, and we are the patients. And with no clinical trials, there are no new breakthroughs. With no new breakthroughs, there are no new medicines. And with no new medicines, there is no hope. And I used to have a boss early on in my career. My boss, whenever I said, I hope this is going to work, or I hope that these guys are going to buy into it, he would always turn back to me and say, hope is not a strategy. What's your strategy? So while we talk about hope and we're being hopeful on breakthroughs and stuff, we have to look at things to do differently. Why do we do this also? We want to get back to the PF community. Part of our involvement in this progression and the disease progressions, we really want to get back to the community. Trials should work with our existing medicines. I've been on my anti-fibrotic medicine for almost 7 years now. I'm not going to give that up to try something that I don't know whether it works or get a placebo. So it has to work with my existing medicines. And then simplify the screening and the engagement process. Many times I fill out a questionnaire and I get knocked back. I get a negative response, negative acceptance of a potential clinical trial. And these are simple questions that we could have been screened out much earlier on within the process. And then simplify the administration, the administering and the monitoring process. My last pulmonary function test was my 26th test that I'd done in 6 years. The first one I did standing up. The second one I did sitting down with a mounted unit. The third one I did, I was sitting down with a handheld unit. Then I had another one that had a monitor with 6 candles. And I had to blow, I keep on blowing until that 6th candle went out. I had another one where I thought she was a sergeant major. It was just, keep going. And I could feel my muscles in my veins in my neck about to explode. So I've spent 45 years looking at taking the variation out of manufacturing processes and business processes, and I look at the FVC result and the PFT test itself, and we have lots of rooms for improvement to look at understanding that variation. I no longer look at individual data points, I look at what that longer term trend is as I plot that out. So, thank you.